show up. Okay, you guys saw my announcement? Yes? Okay, so I'll post your grades later today. Okay, probably tonight. Okay, any questions before we start? Anyone? Questions? Okay, let's wait another couple of minutes. So just bear with me today. I'm still not feeling very well. So, and you guys saw the announcement about the lecture office hours? Yeah? Okay. So I will hold virtual hours next week, Wednesday. So tomorrow. So if you have any questions, you can drop by during that time. All right, so I think we'll get started. Okay, that's not a good start. Anyway, so we'll talk about pox viruses today and papilloma viruses. So we'll talk a bit about HPV, and we'll talk about smallpox as well. Okay, so this has been me since last week. So just bear with me. All right, so starting with pox viruses, they are actually fairly complex viruses, and they have ovoid or brick-shaped particles. So as you can see, the particle here is very ovoid or very oval. It's not spherical, it's not helical, etc. Okay, and you can see the dimensions here, but more importantly, it has surface ridges or tubules which have proteins that can actually bind to the host cell receptors. Okay. Another thing that's curious about these viruses is that they do not have any typical symmetry elements. So they don't have helical or icosahedral symmetry. Okay. And they tend to have an internal core which has the genome, and they also have a protein structure called lateral bodies. So you can see the lateral body here in brown. Same thing here. And in addition to that, there's two different forms that this virus or these virions exist in. One is the mature virus, or MV, and this has one lipid membrane. And on top of that, the second variant is the enveloped virus, or the extracellular virus, which is also called the EV. And this has two lipid membranes instead of one lipid membrane. So you can see the MV virion here and the EV virion here. Okay? So far what we have seen is that these viruses, the shape is different, the symmetry doesn't really exist, and they have a slightly different structure than what we are used to seeing. Another thing to add on is that the name comes from the English word pox, and it refers to the skin lesions that occur on the patients once they are infected with these viruses, okay? So they refer to these blistering skin lesions. There are two subfamilies. There's Cordopox virinae, which infects vertebrates, so things like monkeys, cattle, humans. And in humans, you get variola virus, which is a smallpox virus, vaccinia virus, which is the vaccine strain that's used for smallpox, and another virus called the molluscum contagiosum. This is the only natural pox virus infection in humans, and it's relatively rare. But this is more common in immunocompromised patients, okay? And then the next one is the entomopox virinae, and this basically infects insects, so things like beetles, butterflies, and flies, okay? Now, to add on to this, there are two subfamilies that are divided into several genera, and they infect multiple different types of hosts. So you can see the viruses here, but look at the host range. You have all sorts of animals and humans or the virus that can jump between humans and viruses, okay? And animals, sorry. So you have your reservoirs, but you also have other hosts that it can infect, okay? Now, continuing on, one of the major diseases that was caused by this virus was the smallpox. But it's good to say that it was a debilitating and fatal disease globally. It has been eradicated through vaccination, okay? Because if you actually look at Egyptian mummies from way back when, they still had these smallpox scars. So this tells us that this has existed for a very long time, okay? And when we got to the written records, it suggests that the virus was endemic in Egypt and India by the 1st century AD. And from there on, it spread throughout Asia, Europe, and North Africa by the 10th century, and later colonization spread this even further. So it was spread globally in about 16th to 18th century. So it was a worldwide issue. And in 1700s or 1600s, so 17th century, every child was expected to be exposed to smallpox. Okay, so it was very common, and all children were expected to be exposed to this disease. Okay, and the serious part was that the mortality rate was as high as about 40%. Okay, but in countries where the virus was endemic, the fatality rate was about 25%. Something that shut off. Yeah, it's the vents that got shut off. Okay. All right. So mortality rate as high as 40%, but the countries that the virus was endemic in, it was about 25%. So what does that tell you? Where it's endemic. Why is the mortality rate lower?
Because if the mortality rate is so high that you can't really transmit it to other people.
Okay, so if the mortality rate is high, you can't really transmit to other people. That's true. But why was the mortality lower in this case? Immunity, because those people were exposed to these various strains of smallpox, so they had a better, slightly better immunity. As a result, they had slightly less mortality. And if you look at the disease progression, I don't know why it's showing up like that. Okay, let me put it on a different computer. I don't know why it's not showing up. Sorry guys, 1 minute. Okay, now this computer is taking forever to boot. All right, so continuing on. All right, so if we look at the pox virus disease, what we see is that it's a respiratory infection initially. So it spreads via the nasopharyngeal secretions or scabs, and it has an asymptomatic phase for about 12 days. Okay, so that's the incubation period more or less. And then after that, you get sudden onset of fever, rash, and vomiting. And this lasts for about 4 days. And eventually, it develops into painful rash. The fever will resume, and then the blisters will start covering the body and the airway as well. And as a result, it results in basically your difficulty in breathing and eating because the whole esophagus and the airways are basically full of these blisters. Okay, so it's very, very painful. And you can see these two twins. One was vaccinated against smallpox, and the other one was not. Okay, so you can see the drastic difference that these individuals had as one got the smallpox and the other one did not. Okay, most survivors that do survive smallpox, they do have lifelong scarring. So they do get these pox marks all over their body. Now, since this was fairly deadly disease, there have been vaccination attempts, so to speak, from way back in, I think, 500 AD or even before that. So one aspect I want to introduce you to is variolation. This is an infection which is a non-natural route, and it leads to less severe disease, okay? So this is what eventually led to development of concept of vaccination. And this has eradicated smallpox worldwide, okay? The procedure, the variolation procedure, it was named after the virus that causes smallpox. It's called the variola virus, okay? And the way people used to vaccinate against this is they didn't know what was actually causing the disease, obviously, but they did know that if someone was exposed to smallpox sores or the pustules, they did not develop as severe of a disease, okay? So what they did was they basically they scrape those smallpox sores and then they transferred them to uninfected individuals, okay? So one way to do that was here. You put this long reed in the nose and then you blow on the other end and there will be the smallpox scab on this end. So this gets inside the patient. Okay. Another way to do that is take the pustule from a patient using a needle and then infect an uninfected individual. Okay. So you could scratch the material on their arms. So this is called the cutaneous variolation, or you give it nasally. Okay. And this infection, because it's a non-natural route, is much less severe than the natural infection. And the mortality rate in this case is about 1%, okay? So it was fairly successful. But in late 1700s, Edward Jenner developed a technique for vaccination or immunization, and he was the first to publish that procedure. And in this case, what they did was they took material from cowpox lesion, which is coming from a related virus, and they infected the individuals with those lesions. Because initially they observed that milkmaids that had gotten cowpox did not show any symptoms of smallpox after variolation. So people who are working with cows, they develop these lesions on their hands, for example, for cowpox. But if they were then subsequently exposed to smallpox, they did not develop the disease. And what they did was they tested an inoculation with cowpox from a hand of the milkmaid. So what they did was, again, took those pustules from the cowpox pustules from the milkmaid hands, and they experimented on an 8-year-old boy, okay? So obviously this is a big no-no when it comes to research, okay? But back then, we didn't have those kind of rules. And 6 weeks later, they tried to infect that boy with smallpox through variolation. And luckily, he did not develop the disease. But there's a twist to this story, which is that there were multiple people that actually developed this independently. Jenner was the first one to publish, but again, this seems to have been something that was made up by his biographer to change his image, okay? So even though he published it first, so there is some controversy regarding that. Apparently, Jenner, the whole thing was basically made up in his, biography. But the link between the smallpox and cowpox was first seen by John Fuster. And because what they did was they tried to inoculate a farmer against smallpox, and from there on, they saw that there was no skin reaction. And the farmer then told them that he had cowpox beforehand, okay? So that link, again, shows up, and eventually, basically, what you can say is that the several people did contribute to invention of the vaccines, okay? And by the mid-1800s, the virus that was used to make the smallpox vaccine was changed from cowpox to vaccinia virus, okay? So it has very interesting history. And like I said, smallpox has been eradicated, and this is considered one of the biggest achievements in public health. Just like we saw for COVID, there was a mass effort worldwide to eradicate this disease, okay? So initially, what they used to do was dip the needle in the inactivated virus and scratch it on your skin or put it on your skin, and you get this kind of scar on your arms. Most of you guys don't have that, okay, because you didn't need to. But go talk to your parents, they probably have this kind of mark on their arms, okay? Because that's what smallpox vaccination was. Now, this still remains a subject of intense research, and like I said, the vaccinia virus is now used for smallpox vaccination and in research labs, okay? And for eradication as well as for safety concerns, there's limited worldwide stocks of a live variola virus, okay? So we don't actually keep a lot of this in stock. In 81, there were 4 collaborating centers, US, England, Russia, and South Africa. And in 3 years, England and South Africa removed their stocks. So there are 2 remaining stocks worldwide, okay, of smallpox. CDC in Atlanta and Vector Institute in Russia, okay? Now, problem with this is this virus is highly, highly stable. So even if you leave them in the freezer for decades, these old stocks are still viable. So this virus is fairly resilient when it comes to infections, okay? And if you have time, you can read this article. This was basically, they were cleaning up a freezer in a lab in US, and guess what they found? They found vials of smallpox in the back of the freezer from who knows when, how old, okay? And they had to be obviously destroyed appropriately because you can't just leave these out and just discard them as you wish because the virus will still be active. All right, so let's get to the genome and how this virus works. So it's a linear double-stranded DNA virus. So this is, I think, the first virus that we are seeing that has a linear double-stranded DNA genome. It's fairly large, so anywhere from 150 to about 250 kilobases. The vaccinia virus is about 200 kilobases, and that's what is shown here. And one thing you'll notice is it has these hairpin structures on either end. So these are covalently closed hairpin structures or hairpin ends. As a result, there's no free 3' or 5' ends, and there are 10 kilobase inverted terminal repeats on either end. So as you can see here, so this is your basically the genome. You have your terminal loops on either end. And if you were to zoom in on this area, what you see is several tandem repeats. What are tandem repeats? Yeah. Okay, so it's the specific sequence of nucleotides repeated multiple times. So in this case, some of these nucleotides are repeated. So it's tandem repeats on this end as well as on this end. Now, all these genes, or all this genome basically has about 150 to 250 genes. So you can imagine that this is highly, highly complex, okay? And they encode up to 100 different proteins in the virus itself. And what you'll see is some of the eukaryotic features or some of the genetic features that we see in prokaryotes as well as eukaryotes, So each gene has its own transcriptional promoter. That's generally true for eukaryotic cells, okay? So each of these genes has its own promoter. There are no introns in the genome, so as a result, the mRNA does not need to be spliced. So you can just synthesize mRNA and from there on, you can make the proteins. So since this is a double-stranded DNA virus, where do you think it replicates? In the nucleus, that will be our guess, but that's not the way. That's not what happens, okay? So yes, nucleus is normally where DNA viruses will replicate, but in this case, it has its own separate replication machinery, okay? Now, before we get into the details of that, let's look at the structure and how it's organized. So if you look at the virion itself, it has a biconcave core. So you have the core here. It's concave on both ends, so that's why it's biconcave. And this is flanked by what's called lateral bodies. And these are protein structures that are sensitive to trypsin. So this tells us that there's lots and lots of protein in that structure. But the composition and function of all these proteins is actually not very well known. And obviously, this is hard to study as well because remember, we are dealing with smallpox virus. It's a very dangerous virus. So it becomes hard to study, and we have only so much stock of these viruses. Now, if you look at the internal core, it's surrounded by this core wall structure, as you can see here. And within that, you'll see that the genome itself is in these tubular structures within the core. So that's where the DNA is in this virus. And you can see this tubular structure right here in the electron microscope image. And it has DNA and probably has proteins as well. Okay, that's why there's a question mark here. And it has, one thing that you'll see here is that it has a number of different viral enzymes as well as enzymes required for, or number of different viral enzymes that are required for mRNA synthesis, okay? So basically the virus itself, it packages all those proteins so that you can make mRNA. Now, another thing to add on is that the two versions of the virus, the EV and MV, they differ in their membrane composition because there are differences in morphogenesis of these viruses. So where you find these viruses will determine whether or not you have a single membrane or a double membrane. The MV is mostly intracellular. So it exists within the cell itself. The enveloped virion, the EV, is what gets sent out, and that's what will infect other cells, okay? Thing is, both these forms are infectious, and they use a different set of viral structural proteins to bind to the cellular receptors, okay? So you can see the structural proteins here and here. They are very different. You have this extra membrane in the enveloped virion that has those proteins. All right, so let's look at the mature virion first, and then we'll look at the enveloped virion. So mature virion does have an outer envelope. And this enters the cells by fusion or by endocytosis. Now, again, this is going to attach to the cellular glycosaminoglycans, and this gives it a very, very broad host range. So you can see the extracellular matrix here. This is the cell membrane, and within that you have all these different sugar molecules. So these are polysaccharides that are formed from repeating units of 6 carbon sugars, okay? So you have various sorts of glycosaminoglycans which basically will help, or which the virus will use to attach to the surface of the cells. And these polysaccharides usually have 1 amino sugar. And they are attached to the host cell surface protein. So there are different ones you can see here. Again, don't memorize these names, just understand that the virus proteins, surface proteins, will bind to the various sugars that are present on the cell surface, okay? And this mature variant is actually fairly stable, and it's responsible for person-to-person transmission, okay? Next, we have the extracellular virion or extracellular virus. And this is a mature virion that's wrapped in additional layer of lipid bilayer, okay? And this has its own set of glycoproteins, just like we saw with the MV. Now, this kind of extracellular virus, it enters the cell through phagocytosis, okay? Now, here's your virion binding. It might move laterally. You can either fuse here or you can carry out endocytosis. In this case, you're gonna they usually enter the cell by phagocytosis, okay? And once it comes into contact, or once it's inside the endosome, the outer membrane of this EV will rupture once the pH is lowered in that endosome. And that releases the mature virus particle. From there on, your MV will fuse with the vesicle membrane. Okay, so you basically break open the outer membrane first, and then your MV comes out and it will fuse with the vesicle membrane. And as a result, the core will then get released into the cytoplasm. So it's very similar to what we have seen, except the additional step here is you have first the outer membrane getting ruptured due to decrease in pH, okay? And then the inner membrane fuses with the endosomal membrane, as you can see here. Now, another thing to add on here is that the EV envelope is fairly fragile, okay? It can rupture once it comes into contact with the host cell surface as well, okay? So if it does that, it will release the MV particle into the extracellular space. And this will be responsible for cell-to-cell or tissue-to-tissue spread within an infected person, okay? So two different forms of the virus, slightly different mechanisms as to how to get inside the cell, but at the end, you are going to have that viral core released into the cell. Now, these viruses, like I said, they don't replicate in the nucleus, which is relatively rare for the DNA viruses. They tend to replicate in the cytoplasm, okay? Because what ends up happening is, as you make the DNA copy and then you make all those proteins, all the virus-coded enzymes, they get packaged in the core, and these are the ones that will carry out the early RNA synthesis, as well as packaging of the material. So with the Vaccinia variant, it can actually be considered as a mini nucleus, okay? Sometimes some people call it a nucleoid, okay? And because it itself can synthesize mRNA in that core. And your viral enzyme then transcribe the mRNA, they cap it, they methylate it, and they also polyadenylate that mRNA, okay? So here's the different enzymes that you can see are packaged in these virions, and what the protein is and what the function of that protein is, okay? So you can see that they have an RNA polymerase that's involved in RNA synthesis. They have gene for capping, okay? So guanylyl transferase, that's the cap that gets added onto mRNA. It has a methyltransferase, which methylates the RNA. You have your ATPase, you have your poly-A polymerase that adds the poly-A tail, okay? So there are a number of different enzymes that are encoded by the virus and then are packaged into the core. Because, like I said, you can actually synthesize mRNA within this virion. And there are enzymes that also direct DNA replication. So you have things like DNA polymerase, you have a primase, you have topoisomerase, etc. Okay? So this is involved in DNA replication. So it makes proteins for RNA synthesis, makes proteins for DNA replication as well. Okay? And something very rare, it also has enzymes for nucleotide metabolism as well. Okay? Now, if we look at the genome itself and how the genes are expressed, what we see is that it's actually a transcriptional cascade that's controlled by the various viral transcription factors. Okay? These transcription factors, they bind to specific sequences in viral DNA, so about 35 nucleotides. And what we have is the early transcription factors, the intermediate transcription factors, and the late transcription factors. The early factors are packaged into the virion, and they become active once they reach the cytoplasm. Okay? So these are the early transcription factors that under the cytoplasmic conditions, they will become activated. Then you have your intermediate factors, and these are the ones that trigger intermediate gene expression. Okay? So your VLTF genes actually are controlled by your intermediate transcription factors. VLTF is the late transcription factors. So basically, you make your intermediate transcription factors, and this controls the transcription of your late transcription factors. Okay, so it's all linked. It happens sequentially in a cascade. These late genes then encode, again, your early transcription factors. So it's basically one encoding the other, and then in this case, you're going back to early transcription factors. So they encode these early factors, which are then packaged into the progeny viruses. Okay? And one key thing about this is that this sequential expression is very important because it makes sure that the newly replicated DNA is actually available before you even start synthesizing your structural proteins. So what's the point of synthesizing structural proteins if the genome is not ready? Okay? So this sequential activation will make sure that there is newly replicated DNA, and then you synthesize those structural proteins. Now, the genes that are required for DNA replication are actually the early genes in this virion. Okay? They are expressed immediately after an infection. So here's your MV particle. In this case, it's uncoated here once it binds to the cell. And within your core, you're going to make the early mRNA. Okay? Now, like I said, this is happening within the virus course. So your genes required for DNA replication, they get expressed within the course where you translate these early mRNAs into early viral proteins. So you make the early mRNA and you make the early viral proteins. And these are then involved in things like DNA replication. Okay? And some of these early viral proteins, they actually help dissolve the viral core. They release the viral DNA into the cytoplasm so that further transcription can happen as well as DNA replication can happen. With me so far? Yeah, it's a fairly complex mechanism, so please make sure you guys understand this, okay? Now, we have seen how this replication is initiated or how it gets to the point of replication, but one thing to add here is that you form these viral DNA factories, okay? And these are actually visible under the electron microscope. And the inside of these factories, you have what's called a viroplasm, okay? This is basically whatever components of the virus are, your proteins, your DNA, etc., that's present in these viral factories, okay? So, and here is the nucleus, so this part here is the nucleus, and this is part of the nucleus as well. IV is the immature virion, okay? And you can see the nucleoid within immature virions. It almost looks like a cell. Yes? Almost looks like a cell. But as the virus matures, you can see it getting its characteristic shape, okay? So these are your mature virions, okay? Everyone clear on that? Yes? Okay. Now, how does the genome get replicated and how does it work? Well, what you produce is in these viruses are large DNA concatemers. These are basically multiple genome lengths as we have seen before. So remember we saw concatemers when we were talking about capsid and procapsids and how the concatemer gets cut in the right genome length. It has several genome equivalents. Similar process here, you produce large DNA concatemers and then you later resolve them into monomers, okay? So how does this process work? So this is your poxvirus DNA, double-stranded molecule. But remember that this has closed ends. It does not have open ends, okay? So you're forming these closed end structures, and that's how the DNA exists in this virus. Now, first step is a host cell enzyme called helicase, which will nick the DNA on one side, okay, of this molecule. Okay, so you make this small nick. And remember we talked about repeated structures here and here on either end? Yes? So these repeated structures, these terminal repeats, they will actually fold over and form hydrogen bonds between themselves, okay? Because these sequences are complementary, because they're repeated structures, you're going to form these hairpin loops on your DNA, okay? So first you snip or cut one end, and then as it unwinds, some of those repeats will base pair with each other, okay? And this part here, the circle here, indicates the 5' end of the genome. All right, now you have these hairpin structures. Well, guess what? Our DNA polymerase needs a primer to make new or to replicate DNA. So these hairpins actually act as primers, okay? This is called self-priming, okay? So this genome is making its own primers because of these repeated structures, and DNA polymerase will happily go in and then start synthesis on one end of the strand. DNA synthesis will continue, and as it continues throughout the molecule, on the other, when the DNA polymerase reaches the other end, the other closed end, it's going to basically loop back and synthesize this way, okay? So you have this kind of structure, and this part where it bends, that's the concatemer junction. That's called the concatemer junction, okay? So your polymerase will continue happily along. It will read the template, and it's going to start adding the nucleotides. Once it reaches this area, it's going to basically continue synthesizing the genome. So now what you have is 2 copies of the genome, okay? One on each end. So how do we fix this? Because what you see is you get these cruciform structures, which are the concatemer junctions, because the process continues on with self-priming, and it produces a concatemer or 4 genomes or more, okay? So as the DNA gets replicated over and over, you're going to form these cross-like or cruciform structures, okay? And then an enzyme comes in called resolvase. That resolvase basically resolves your concatamers. It basically recognizes these cruciform structures where the concatamer junctions were, and it will digest the genome in those structures, okay? This resolvase is also encoded by the viral genome. And as you make individual molecules, you are going to ligate or join the ends together. So you seal that hairpin structure, okay? And the two strands are now bonded together, and you form your terminal loops for the genome, okay? So you carry out DNA replication, you continue on through the hairpin, okay? And you do multiple of those, and then resolvase comes in and cuts those different concatamers into individual genomes, okay? Everyone clear? Yes or no? Okay, take me through the steps. Step 1. Okay, so helicase will nick one of the closed ends, which happens close to the 5' end on the molecule. Then what?
The terminal repeats kind of like they flip over and hydrogen bond with each other.
Okay, the terminal repeats will form the hairpins. They will hydrogen bond with each other. Then what? Let's pick someone from this side. Who wants to tell me? Okay, your DNA polymerase uses the hairpins as a primer. It will synthesize new strand of DNA. Then what? What's next? Someone from here. Who wants to tell me? What's next? Someone, come on. You just heard it, so let's hear it back. Anyone? Okay, no one's interested. How about here? Anyone? Okay. Yeah.
So the polymerase keeps going until it reaches the junction, and then it kind of loops back.
Okay, the polymerase is going to continue synthesizing until it reaches the junction, and then it loops back. And then using those primers, it's going to synthesize more strands, okay? Or it's going to synthesize multiple of these concatamers, okay? Once you form these concatamers, they have this cruciform extrusions, which get recognized by resolvase, and then that cuts the genome into individual, or that cuts the DNA into individual genomes, okay? All right, so here's the steps for the whole process. So you have a site-specific nick in one of the hairpins on one end, okay? So this figure is a little bit more detailed than what we just saw. So that's the basic concept, but one step to add in here is that once you nick this fragment, you need an extension on the 3' end, okay? So this extension is about 100 nucleotides, okay? So you cut here, this whole molecule will open. Now there's a gap left here. So what you need is a synthesis from this strand in that direction. And after this extension happens, your hairpins are refolded back. So this is where you form those primer structures, okay? And from here, you can use this for further DNA synthesis, and then you synthesize the entire genome length with that DNA polymerase, okay? So it's going to synthesize in this direction. It's going to synthesize the entire genome length, and as it loops back through the second hairpin, it's going to generate a head-to-head dimer, okay? So it's going to generate a dimer of the DNA molecule. Finally, you need to ligate these two fragments together. So ligation will happen here, and you'll get the full molecule, okay? And like I said, Once you do this, your concatenamers need to be resolved. In this case, it's only showing you 2 molecules, but like we saw, it can have 4 or more. So resolvase comes in, it makes a single-stranded breaks in the ends of the concatenamer junctions, which you can see from the previous slide. So again, you resolve these concatenamers, and you form these DNA molecules with hairpins, okay? And then these hairpins get joined together by ligation, so you seal the ends for this DNA molecule as well, okay? All right, let's take a break, and then we'll continue in 10 minutes.